I'm pleased to welcome Ana Caetano from Queen Mary University of London. Ana authored a paper in the JDR special issue on imaging that describes different computational approaches to bioimage analysis. Hello, Ana. Could you describe in general terms the major types of bioimage analysis that you covered in the paper?
Hello, Daniel. There are essentially two types of spatial transcriptomics. So on the one hand, we have a targeted approach where we can have a selected or curated number of genes that we can analyze. And then we have untargeted approaches or whole transcriptome analysis where we can profile more or less everything that is in the tissue. In terms of comparing both methods, essentially they differ in terms of cellular resolution and transcriptomic coverage. So for example, methods using fluorescent in situ hybridization, they can offer a high resolution imaging, but with a pre-designed probe list, so not unbiased, whereas the whole transcriptome, we can while it's not single cell resolution, we can have a complete readout of all the genes in the tissue. So it really depends on the research question we have.
Okay, thank you. Given the fact that the probe-based approach has several thousand genes now, and issues with depth of sequencing related to the transcription approach, is there a particular reason to select one over the other?
I think the one, so the target approach is the one that we have a panel list of genes. It's useful when you have a lot of information from the tissue before, so when you have a hypothesis. Whereas the other one is more on the discovery side, so when you don't know the identity of rare or subtle cell states or cell types that you want to identify.
Okay, thank you. What were some of the critical advances that were needed to progress from single gene in situ hybridization to transcriptome-based spatial analysis?
So there are already some single cell in situ hybridization moving to whole transcriptome profiling, which I think will be available from next year. So this is, again, particularly useful to discover new and rare cell types that have been missing out from single cell datasets in low resolution spatial transcriptomics. So it's such a fast-pacing field that I feel like our review is probably already outdated. So I'm looking forward for next year and to have these technologies out.
In the manuscript you co-authored with Alicia James and Paul Sharp, you focused on three areas: image restoration, segmentation, and tracking. Why did you choose these areas?
So essentially these areas are the most common cell imaging or imaging tools that can be applied to extract quantitative data from an image. So for example, we have image restoration, which means essentially denoising the image, so making the image more sharp. So when we have high objectives, it's often difficult to get a nice image. Segmentation, for example, when we want to quantify cell numbers, so we can segment cell nuclei automatically in order to have an unbiased quantitation of our image. And then tracking, it's important, for example, in live imaging, where we want to track the beginning and end of a trajectory of a cell. So those are basic analysis tools that we can apply to our research questions.
Thank you. What key insights have you gained specifically in your own work using spatial transcriptomics?
So I think for our work, which is on the understanding of oral mucosa biology, we gain a novel understanding of particular cell states in health and periodontitis that were previously unrecognized. So initially, we used spatial transcriptomics to build on our single-cell RNA sequencing studies and as a form of validation as well. but we were able to discover much more. For example, we applied deconvolution spatial analysis that allows us to detect the 3 different types of epithelia that make up the gingiva without any prior information or data training. So we could have a complete readout of these epithelia regions and study how position determines oral epithelia cell fate or identity. We were also able to study fibroblasts in more detail, which are particularly difficult to study in space due to the lack of good markers. So we found these inflammatory fibroblasts near the junctional epithelium with a unique gene expression profile. So we hypothesize now that this may be a potential survival niche for immune cells, leading the way from innate to adaptive responses. We were also able to model candidate cell-cell communication networks in space and find potential patterns unique to periodontitis. So I think there's still a lot of work to be done with this data, but I think it has the potential to answer and generate a lot more questions.
And on a personal level, what advice would you give to a young dentist considering a research career?
It's very difficult to give advice, but I'll do my best.
It's easy to give advice. It's hard. It's hard to follow it.
That's also very true. So I'm going to speak from my experience, but and I think it might be a bit cliche, but I think it's true. So I think it's find or follow a question that is intellectually challenging and that excites you, and follow that. I think this may happen more easily if you are lucky to find a lab or a supervisor that gives you the freedom to explore and ask questions, that encourages you and maybe sees your potential. I guess also read widely, don't stick to your fields and be open to learn and get better. So I think clinical academics are an odd species because we are in between clinicians and scientists, so it's not the most comfortable position at times, but I think resilience and surrounding yourself with good people, that helps.
Mm-hmm. And as your career has developed, you started as a student, and then you've done a postdoc, and now you have a faculty position. What are a few things that you really enjoyed in this early aspect of your career?
I have a few favorite things because I'm enjoying it. So I think setting up the lab from scratch, that was very exciting. My first microscope, so that was very, very a very nice day. And building up a team. I think since I have a research assistant and now the PhD student starting, I think having those daily interactions and just having fun in a lab, that's the most enjoyable part of the job.
Yeah, I would agree with that. My own experience is that the interactions between researchers is very interesting and rewarding. I think that's what you're saying, and that's my own experience as well.
Definitely true.
Well, it's been a pleasure, Ana.
You.
Your career is certainly on a great trajectory, and I wish you the best.
Thank you so much. Thank you very much.